The overall goal of this procedure is to rescue Recombinant Newcastle Disease Virus, or NDV, from cloned DNA. This is accomplished by first infecting mammalian cells with a helper virus encoding T7 polymerase, and then transfecting those cells with plasmids encoding the minimal replication machinery of the virus under control of the T7 promoter. The second step is to co-cultivate the transfected mammalian cells with avian cells, which are from the natural hosts of the virus. The newly generated virions released from the mammalian cells will infect the avian cells for a first round of amplification. Next, the supernatant of the co-culture is inoculated into embryonated chicken eggs for further growth of the rescued virus. The final step is to collect the allantoic fluid from the inoculated eggs. Ultimately, a simple hemagglutination assay is used to show the presence of NDV in the allantoic fluid and the success of the rescue process.
Recombinant viruses rescued by this method can provide insights into different factors affecting infection and can also be used as vectors for the delivery of antigens for vaccination and antitumoral therapy.
To begin, split mammalian Hep2 or A549 cells the day before transfection in 6 well plates. The density of the cells should reach 80 to 90% confluence the following day. For each virus to be rescued, include 2 to 4 different wells. Also include 2 extra wells for the controls PCAGs GFP